Okay. All right, thank you very much for coming. Today, we are very excited to have Dr. Yingqi Zhao from Therapeutic Cancer Center. Yes, the floor is yours. Welcome.
Yeah, thanks, Yingqi, for inviting me and for the introduction. And it's my first time to be in Ann Arbor, so I'm very excited to meet new and old friends. And yeah, and I also have my client mentor in the audience, makes me feel nervous. Yeah. Today I'm gonna talk about some of our recent developments on how we address real world challenges in cancer early detection and therapeutic studies. So particularly, I'll try to cover two topics. And the first one is on how we can design a cancer randomized screening trials. And the second is how we can better evaluate treatment effects, leveraging both randomized trial data and perhaps some real world data. And the first one is on this cancer screening clinical trials. In the session, like coffee session, we were just chatting about all these recent developments in cancer screening tests that were currently on the market. There were commercially available tests that can actually test whether you are likely to have elevated risk for a basket of cancers. And to evaluate those tests, usually it's different from our therapeutic test, right? The population usually is asymptomatic, it's more healthy, it's healthy population. And you want to see after all this sequence of actions, are you gonna really see the effects of this screening test? So unlike a therapeutic trial, that if you're designing a screening trial, you're starting from healthy population, and you want to follow them for a very long time to see whether this test will really lead to some effects, right? So if you want to calculate, let's say, how much, and to hypothesize, to power the study, you want to know how much effect this screening test will lead to. It's not like a therapeutic trial that you can run a small study and to see the effects. Because even if you run a 50 trial with 50 people, you still need to follow them for maybe like more than 10 years. So it's not practical. So there are several questions, like to answer this Q1 question, will this screening test really have effects in terms of either like to change the stage distribution, that you can see less late stage patients in the end, or if you do see the mortality benefit, that you can reduce mortality in the end. So to see such effects, you need to carefully design the study. And the second is that how to evaluate this screening test that you're willing to do a trial, right? The screening test probably is usually summarized in terms of sensitivity, specificity. This kind of test statistics is like say 90% sensitivity with 30% false positive rate. Is that good enough? So that's also something we want to evaluate. And the third is, what are the harms associated with screening tests and its follow-up procedure? Like say the screening test itself is fine, but the thing is the follow-up actions could be harmful. That if say you see a positive results and then you need to go through all those workup procedures, that's harmful. And if you see a negative results, that may also lead to harmful, that you may skip regular screening. And those are the things we also want to evaluate. So there are a lot of issues here. And in my talk today, I'll just focusing on the first one. We want to develop a trial such that with this trial design, we will be able to answer this question. Does the screening test can change the distribution of cancer stage? So as I said, for cancer screening trials, it's usually different from a therapeutic trial. It's usually very large, very long, like some large trials like national lung screening trial is more than 50,000 with a long, more than 5 years of follow-up. And to really see the impact, the mortality endpoint is usually the gold standard. It's still treated or viewed as the gold standard endpoints. But to see that type of impact, it will require even longer follow-up. So recently, people have been interested in looking at this late-stage cancer incidence to kind of look more like intermediate endpoints before the mortality. And then our goal here is to provide a statistical method to design the trial with late-stage cancer incidence as the primary endpoint. And the problem, as I said, you cannot run a preliminary trial just to evaluate the effects. What you have is your screening test properties. And you can look at prevalence, your incidence, and you need to use all this information, try to project what would happen, what the effect size might be for this screening test. So for a randomized screening trial, patients are randomized to control, which is whatever standard of care is currently, or we can go through a sequence of screening. And then in the end, we want to check whether there is effects, right? Instead of mortality reduction, we can check whether there is a reduction in late-stage cancer incidence. That can give us like a shorter, like you can see the results earlier, and which is also considered as it's a good impact that very highly correlated with mortality reduction. So essentially, the hypothesis is that just this long hypothesis is the screening wouldn't reduce the late-stage cancer incidence. And problem again is how we can specify the effect size such that we can design the study. And of course, there were other issues, like say, how many round of tests do you want to implement, and how long the follow-up you want to do in order to observe that late-stage incidence reduction. So we leverage these multi-state models for cancer screening. And the classic model is a 3-state model, which there were cancer-free normal state, and then they can transit to preclinical state, and then transit to clinical state. And this 3-state model has been used, like say, to design the NLST, the National Lung Cancer Screening Trials. And in that trial, the mortality was used as an endpoint. But in order to look at the late stage reduction, we need to differentiate the early stage and the late stage in this model. So we use this expanded model, which include 5 states. So for preclinical, there were early state, preclinical state, and late state preclinical state. So when we say preclinical, means that it's not symptomatic, but if you use a screening test, you would be able to find that, okay? And for clinical state means that it's symptomatic and you would be diagnosed just from the symptom. And so there were 5 states here that you go through early preclinical and you can directly go to early clinical, or you can go through late preclinical and finally becomes late clinical. And for the model, we consider different set of parameters. The first one is, as we said, the parameters for screening program that usually we will be able to have the sensitivity for detecting cancers in early preclinical and in late preclinical. Of course, we will see higher sensitivity when you are in late preclinical, but but lower sensitivity for beta E. And this can be obtained usually from earlier phase of biomarker studies. And then again, we need to also determine the frequency and timing of repeated tests and analysis time. So that's also part of the model and part of the design. And then the second set of parameters are natural history. And we need to look at how fast the cases will move between this state. That the first one is how fast you are getting, like population-wise, will move into this early stage preclinical cancer. And we make assumptions that the time spent in each state is exponentially distributed. And the surgeon time, mean surgeon time is essentially the duration, like how long this will stay in the state. And we consider this stable disease condition, which means it's a constant, like the rate is constant. And we show that the relationship, actually the incidence and the prevalence and the mean surgeon time, we can actually have this relationship. And this bathtub analogy is kind of, we can use this to understand what does this stable disease condition mean, and what does, like how come we get these quantities. So stable, so we can treat this bathtub, their count pool is like a prevalence, we can see as prevalence pool. And the inflow and outflow incidence, inflow is like they change into this state. Outflow flow means they get out of this state. The stable disease condition essentially means how fast this inflow is the same as how fast you get out. Okay, so inflow is outflow. And then then this prevalence is essentially is this incidence times the duration, how long you stay in this state. Okay, that's how we get these numbers. So the mean surgeon times and with exponential distribution assumption. And all these incidence parameters can be obtained from like SEER data and some other sources. And now with all those parameters specified, we can derive the probability of observing late-stage cancers. So essentially, this we observe late-stage cancers either by screening detected that you detect using screen tests in that late-stage preclinical state, or it's an interval case that it's not screen detected, but later on showed up with symptoms and was found. So we separately derived the probability of late-stage cancers by screening test and the probability of interval cancer. So I wouldn't get into details, but essentially we need to list out all the possibility of getting detected by this round of tests. And then these quantities can be written as a function of those parameters we specified. And then once we have that, so I would say this is the most kind of involved term. Once we have that, then it's going to be a standard practice, like we derive the effect size and derive the power and determine the sample size. So to validate this model, we try to redesign the national non-screening trials. So it was, as I said, it was a practice-changing trial that after this trial, the low-dose CD was established as a standard of care. And it randomized eligible smokers to chest radiography and low-dose CT. And the Hu and Zeller model, which rely on that classic three-stage model, was used to design the trial. And they use cancer-specific mortality as the endpoint and look at the reduction in mortality at year 6. So we redesigned this trial with late-stage cancer incidence as the endpoint and using the same set of parameters and with some additional inputs for early and late stages, like say sensitivities, and we considered a study design with 3 annual tests. And we can see that with at the same time of year 6, we can use less participants. We only need 40,000, okay, to detect this 20% incidence reduction. So that's also what's observed in the trial. And these crosses are what we projected. And these crosses are what was observed in the trial. And these lines are what we projected over time. So I think it's close that we can kind of try to get the model calibrated. Yeah. And now, so that's our first kind of work, that the first step towards this set of questions. And what's next? What we're currently working on is to look at the cancer-specific mortality. So not only late-stage reduction, because it's so first, it's not a gold standard outcome of interest. And what we want to do is trying to kind of use this type of model, trying to connect these two endpoints and see how late-stage incidence mortality related to the mortality. And what we did is to extend this 5-step model to incorporate the mortality endpoint to look at the follow-up survival after the screening. And we characterize different paths, trying to account for some biases that we might observe in screening. Especially there is lead time bias, which means that we usually would observe a better survival, even if it's the same stage, right? They compare screen detected versus the clinically detected interval cases. But that could be lead time bias because you detect them early, and if you are counting from the time of detection to death, that's so maybe your survival is not changed if it's just because you detect them early. So that's one possibility. And the other possibility is called length time bias, which means that those slow progressed cancer are more likely to be detected. So you see a better survival for those cases. So what we're trying to do here is trying to characterize different paths that are essentially, like for example, this path 1 is what would have been, like early clinical, but was intercepted by the cancer test, and what would be the original, the survival rate from the original diagnosis that would have been. So that's it's not from what the cancer time detected, it's not from time of detection, but we need to consider this kind of potential outcomes, like what would have been if there were no detection and the original time diagnosis. So we laid out this 5 paths and trying to again model the impact on the death. So what?
First question. So I see lambda 2 and lambda 3 and lambda 1. So does the screening change the values of those parameters? Is that how you? What's the effect of screening on the?
So screening does not change these parameters. These are related to disease characteristics. Screening is the sensitivity of those data. That's the screening test characteristics. And those tests will, those betas, those sensitivity will mainly change the reduction in late-stage cancer incidence. But how that will further change the mortality will depend on, like, say, the therapeutic options, how good it is to treat an early stage cancer patients. Like is it really will change the survival depends on like how good an early stage patient will get treated.
So someone who has screening, if they enter this picture, they don't go through the lambda-2 arrow, they go somewhere else in the picture. Is that sort of?
So you mean go through lambda-2, like go to the preclinical?
Do you prevent that, do you?
Yeah, so if they're detected here, then they will not further move into LP. So if they're detected, right? So they could either move to LP or they could also move to early clinical case, like they do not further progress to late stage, but just have symptoms manifested in an early stage. So that's two possible paths when they reach early preclinical. And then the final and the other questions are more related not only efficacy, but also the harm associated with screening test. So that's also something we need to characterize, and harm includes false positives, right? And also overdiagnosis is another type of harms. That's also that's something like we haven't get into. It requires to consider like computing risks and other modeling type thing. Like we haven't done that, but that's definitely something we should consider when evaluating such tests. Yeah. So essentially, our work is trying to characterize the benefit and harms in screening trials, and to design such trials, it requires understanding of the stage-specific biomarker performance and also disease natural history and how they kind of interacted with each other. So yeah, I think I learned a lot from this project that I didn't have experiences with such models before. But I think it's an important things to consider, like especially with the current need in evaluating such tests. So, and I think in the future it's also like when we decide whether to take such tests, we also need to understand the benefit and harms of such tests for our own sake of decision making. So, any questions for this? Okay. Yes.
Just a clarifying question: how are the patients like determined whether they're recruited or not?
I think it's on volunteer basis, right? Like usually it's patients participants will see information and they see if they're willing to come to this test. And so now I think for the screening field, it's some biotech companies, they've already started to run big trials in UK, right? And NCI is kind of late in this game, and they started to establish this network and trying to do a trial that's more like a feasibility vanguard trial and see if this can really be done in United States. And so I think like it's more like just to see if we can get patients. That may be something you related to your question, like can we really get patients? And if we're getting our patients, are they really doing this test? So yeah, that's an ongoing study. Yeah. And Hutch is coordinating that study.
Yeah. So I have a naive question. So one of your endpoints is to compare sort of incidence rates, right, between the two arms. So if you do more screening, isn't that obvious that you're gonna have more incidence because you do more screening?
Yeah, so that's why we use late stage incidence. So the goal is to reduce the late stage incidence. What you said is related is like overdiagnosis. You do more screening, usually you see more cancer cases. So I think there were studies like using cancer detection rate as an endpoint, which we think it's not a good endpoint because we are likely to see an increase in cancer detection rate, but that might be just overdiagnosis or, like some cancers, even if you see many early stage, you may see a large increase in early stage cancers, which may never be found. So that's an important thing. Like when we choose the endpoint, late stage cancer incidence is considered that we wouldn't kind of have this overdiagnosis issue. But the problem again is like even if you see effects in late stage incidence reduction, it still may not translate into mortality benefit. And there were trials that actually see effects in incidence, late stage incidence, but they didn't show mortality benefit. So yeah.
So just a follow-up question. So if a patient is detected at early stage, will they be treated or?
Yeah, they will be treated.
So that the hypothesis that if they're under treatment, then they're gonna either delay their progression to a late stage or never enter the late stage.
So that's after, right? That's not related to what endpoint we're using. We're just using like what stage they are detected, they are at the detection. So what's happening afterwards is related then to mortality. Like,
I see. So once they are detected,
well, once they're detected, we look at their stage instance. Sorry, look at their stage and count
Can you talk about the sensitivity of the test in terms of the disease stage, right? Because if you're late stage, maybe it's not easy to detect than it's early stage.
No, yeah, it's
your-
the other
your
way.
performance-
If you're in early stage, it's harder to detect.
Right, I mean, the other way. So your performance of, because depending on where you implement the test or rate, it's gonna be different, right? What's the impact of that on your design?
Yeah, I think it's, so that's why like, we want to kind of see like what kind of sensitivity is sufficient, right?
It's not in your equation, right? Is it in your equation?
It is, yeah, it is somewhere hiding,
Okay.
hiding in these quantities. It'll show, because that's like say, because when we check like whether there will be a case, it depends like at this run, are you gonna detect it or not? So if there is truly a case, are you gonna detect it? And that's related to the sensitivity. And then if, and you might miss, right? Like then next time, are you gonna detect it? So there is a series of calculation that needs to account for the possibility of missing that case or, yeah.
So I have a relevant question. So for the screen, you're showing that you have the same technology to the screen. I mean, in practice, people may use different, some may have better health insurance. They can go more biomarkers in the screening, but some people may have that level of, health insurance. They can do like lower level screening, like, so I don't know, like cancer screening itself that could involve different technology, the screening or different biomarkers they use, right? Depends on
Right.
actually practice. You shown here that this screening is the same.
So, yeah.
In terms of sensitivity, of course.
Yeah, it's different arms, you may have different tests, right? Like usually control arm is just whatever regular practice is, and screening arm would be whatever regular practice is plus this new test.
Okay.
And
I
I think you when we consider that sensitivity, like it's gonna be like a combination, it's a package. So it's not just this, yeah, so we need to, so it's not possible that we have a test and then we are not doing mammogram, right? So it's always a package that we need to assess as a whole. Yeah. Okay. And the second problem I'm gonna talk about is how we can kind of better use the clinical trials data to combine that with some real world data, trying to kind of reduce the cost of follow-up. And so what currently, as I think as recently there have been a focus or like a push for pragmatic trials, right? So the pragmatic can be, and like there are a lot of aspects that could be considered pragmatic. Like I talked to Yingqi, that cluster randomized trial is one way to reduce cost, especially for screening trials. But there are also other set, like we're hoping to reduce patients' burden, like we try to reduce the number of visits, and we broaden the eligibility criteria, right? Like we try to streamline the data collection, and we want to minimize the cost. And one thing we want to do, or like within the SWOG, the National Cancer Trial Network Group, and what we want to do is trying to augment the trial with some real world data, and such that we don't need to collect as much in the trial, but get more data elements from this real world data augmentation. And one example is this prostate cancer prevention trial. It was launched to examine whether finasteride can prevent the development of prostate cancer. And the trial has a 7-year follow-up, and it just cannot continue due to financial and logistic consideration. And particularly for this trial, the result was very controversial. The trial results showed that the finasteride could reduce the prostate cancer incidence, but will increase the high-grade cancer incidence. So the trial was not, the drug was not approved for the purpose of preventing prostate cancer. But so there were like a lot of work trying to understand like why this happened. And one thing is to really trying to see the long-term effects of this drug. So beyond 7 years, like are those really like impacting patients, like those high-grade cancer? So what we did was, because considering that prostate cancer is mostly like occurred in all male populations, and this trial, when it's ended, most people will enter this 65, right? Like the eligibility for Medicare. So the SWOG group actually linked the participants' Medicare claims data to their clinical trial records. So based on their social security numbers. And after this linkage, the follow-up time for participants was prolonged. So we can just grab those events, like what's happening after the trial from the Medicare data without just running this positive trial. And the prostate cancer and death diagnosed after clinical trial can therefore be identified. So then with this kind of combined trial and linked with Medicare data, we can now like evaluate long-term effects, not only limited to that 7 years, but beyond 7 years, like we can also see what's happening. But there are also some issues for this data linkage. So first, the incorrect linkage can happen, right, due to mismatch. And then the other thing is incomplete linkage. So first, not every participant maybe reach the Medicare age, right? And by the end of clinical trial, and also participants may not be willing to share their social, so you don't get to link them. So that's that cost that leads to this incomplete linkage. And especially, so for PCPT, it is fine. Like we have 70% get linked. But for some later trials, the linkage rate is much lower. So for this work, we assumed that the unique identifier exists, and we only look at the incomplete linkage problem. So we assume, so for this long-term treatment effects, we use the Cox model to define this long-term treatment effects. And this is assuming that every participants are linked, right? So that's, we can, if everybody is linked, then we can run this model and evaluate this long-term treatment effects. And the parameter for this A, the beta 1, is the long-term effects. And for particularly, we consider this change point focus model. That's due to the trial design for the PCPT. So for this trial, the complication is that they did an end of study biopsy, right? So patients during the study will be diagnosed with prostate cancer if they have symptoms and they do biopsy, and they want to biopsy everyone in the end, just so that they do not miss cancer cases. So for this study, we would see a bump in prostate cancer cases in the end because everyone gets biopsied. So we will see a bump. That's why we add this change point at the study end so that we can account for this sudden bump. We add 2 change point because after the study end, then the incidence rate again decrease. So this work is with Gong, a student from Biostatistics, and Yingqi and Zhao, our colleagues. The model specification, again, because we use Cox model to define the long-term effects, then there might be some model misspecification, but even if Cox model is still misspecified, this parameter is still well defined. And I think go with Cox model is also because it's still widely used and well accepted by collaborators. So now we can look at this data linkage issues. So it essentially will categorize participants into different categories due to whether they are linked or whether they are censored in the trial, right? So for those, so here in our study, the failure is the cancer, like either cancer, developing a prostate cancer or the death, okay? So the case 1 is they are always linked. So regardless they are censored or not, they further on they were linked. So we can see that the plus is that even if they are censored in the trial, they can further, that if the Q equals to 1 means they're censored in the trial, but later on they get back, like we can get more information for this set of participants. And the case 2 are those who are censored, who have events in the trial, right? And the case 3 are those who do not have events, but then also not linked to the follow-up data set. So yeah, these first participants are those who have events in the trial, but then also later on was linked to the Medicare data. And this case 1 is what we call complete cases, that we just look at the set of participants who have linkage data. And that's the kind of the baseline method we want to compare to, right? So we just look at this complete case data. And the second participants are those who are censored in the trial, but then later on, we link that participants in the Medicare, and that gives us more events, right? So that's what we like. Like we get more participants events data. And for case 2 are those who are not linked but just censored, but have an event in the trial. So for this, we also have the information. And for the last case are those who are censored but also do not have follow-up data. So as I said, this case 1 are the complete case analysis. So we only include participants from case 1 who have the linkage data. And we can also, let's say, oh, this case 2 participants, they have the events in the trial. Why not just add them to the complete set? But it turns out this set of patients, like by simply combining these two, will be biased because you simply adding this case 2, well, you kind of your inclusion of participants will depend on their outcome, right? Like they have events happening in the trial. So that's two methods. Yeah, and then we consider 2 methods for such scenario. So first, so essentially the first one is called non-censored, non-linked-as-censored. So that's the first method we proposed, and the second is an weighting methods that we developed the ways such that the estimating equation is unbiased for the full estimating equation. So what we did is just write down the likelihood, assuming everyone is linked. And then, however, we do not observe the times and delta, the sensor indicator for participants in class 3. So that's where we need to kind of weight. Essentially what we did is trying to weight this portion to represent the whole so that we can make up the missing information from case 3. So that's the weighting method. And this non-linked-as-censored method is just treating this set of patients as censored and set their times at when they were censored in the trial and then fit the Cox regression. And such method expect to perform well when model is correctly specified, as long as the censoring time is independent. There is a conditional independence. And then the other method, the weighting method, is relying on this linkage at random assumption that whether you're linked just depend on your covariance, right? And among those who are censored in the trial. So yeah, because we want to use this set of participants to represent this. So it's just looking at those who are censored set and consider this assumption, linkage at random assumption, and then develop the ways that essentially these 2 cases, these 2 are giving weight 1 and case 3 are weight 0, but this part are inverse weighted by the linkage. And then we develop the IPLW weighted partial likelihood. And we also develop the augmented IPLW weighted method. So this augmentation is a bit complicated because it has 3 different functions that if you want to kind of make sure all 3 are correct, it's not that easy. So we derived it for completeness, but for implementation, we actually went with the IPLW method. And sometimes I think we can still play with augmented IPLW method, but it's not that simple to kind of correctly specify all the components in this method. So for the study, this PCPD Medicare linkage study randomly assigned 18,000 men in the mid-90s. And we exclude more than like about 2,000 participants who have a gap between the end of trials and the start of Medicare claims. So that means they have not turned to 65 by the end of the study. And after a few years, they get enrolled. So we excluded these participants. The median follow-up after the linkage is 16 years. Okay, so it's much longer than the original 7 years follow-up. And among those 16,000, we have more than 70% were linked. And we have about 2% who have the events during the trial that were not linked. And we have 25% who were censored, but then later on, who are censored who did not have a subsequent linkage. So that's where we do not have information. And the outcome is time to diagnosis or death, whichever comes first. And we fit Cox regression with covariates like PSA and some other demographics. And we considered additional covariates for logistic regression to model the linkage, okay? Especially this education level, which may affect the willingness of participants to link for the linkage. And we add 2 change points at 6.5 and 7.5. That's where the kind of end of biopsy started, happened during this period. So during this period, we see a steep increment in the cumulative incidence, but then it goes back down. So that's why we added 2 change points. So the results were compared across different methods, NLS, AC, IPLW, and complete case and complete case plus. So I think in this case, most are similar. It may show that Cox model is correctly specified in this case. And yeah, but in our simulation, we see that especially the complete case plus is very susceptible to bias, because as again, the inclusion of participants rely on the cancer events. And that's the end of my talk. So this work, both work are supported by NIH funding and PCORI funds. And I want to say that we're hiring. The institute is hiring for faculty, and I'm also hiring for postdoc. So if you're interested, welcome to Hutch. It's a very nice place to work. And if you have any questions, yeah, you can send emails. And yeah, I forgot to mention that the first work is a joint work with Ke Hao, who was a student in Biostat, UW, and Yingqi, who is a colleague, and Ruth, also a colleague of mine. Thank you.
Questions?
Once you link to Medicare data, it's observational data, right? So do you need to worry about any potential confounding in the linkage?
So because it's the study itself is randomized. So yeah, from that perspective, that's still preserved, right? If you're looking at long-term
You
effects.
have somebody not linked, so it could be different person.
Yeah.
And also could be like, say, after trial, people get a different drug, different medication.
Yeah.
All these things can be.
Right, those are, yeah, those could be the issue that, if there are, definitely like need to account for that if they say they're switch, to another treatment, and yeah, that's different. I would say here we just still just look at their initial treatment, but I agree that could be and the poor linked are different from poor non-linked, that's also will be an issue. But here we have this linkage and random assumption that kind of get, like trying to say that you can use the covariates to adjust for the potential differences. But if there are unmeasured issues, then yeah.
I have a question. Can you provide the intuition, like how those case 2 and case 3 people, because they don't have the long-term outcome, right? How can they help with the long-term effect estimation, or just based on the case 1 analysis?
Yeah, so for case 2, they already have events, so that's already happened. That happens. It's just like if with 7 years of follow-up, some patients we still haven't get events, right? Just with longer follow-up, we will have events. But as I said, you cannot just simply adding those who have events all together because this case 2 are those who have shorter events, and they're just, if you're just including them because they have these events, then that'll bias the results.
It's specific to survival analysis.
Yeah.
So I'm curious about the covariates you used in the Cox model. So all the covariates only come from covariates collected from the trial. So no covariates from the Medicare data.
Right. Yeah.
So if someone after the end of the trial received certain treatments or something else that were captured in the Medicare data, would you feel that there would be something that should be adjusted for?
Yeah, I think as a like follow-up work, I think that should be something to consider, especially if it's especially now, like cancer immunotherapy is so common, right? And for those who for earlier trial, it's not, they say if we were comparing immunotherapy versus chemotherapy. After linkage, those who undergone chemotherapy, those patients will definitely get immunotherapy. So I would say in that case, such comparison may not be convincing because you're not accounting for what's happening after the those treatment practice change could very much affect the
So for the diagnosis that in the Medicare database, you don't know exactly when disease occurs. When they come to the clinic for diagnosis, probably already occurred. I mean, but a while ago, I think it's more like interval censoring or current status data. I mean, clinical trial probably is easier because you have frequently scheduled visit, probably roughly when that happens, right? So, but for the Medicare data, I would say that you don't know exactly when that happens, but of course that when the patient come to clinic to have diagnosis, you only know at that point that disease occurred, that you don't know when exactly.
That's a very good point. That's actually what we do here is very simple. Like we just take whatever that gives us. But I agree that Medicare data, the diagnosis is not perfect. Like there might be wrong. They just rely on coding, like those codings trying. So my colleague, like Joe, like he actually examined different types of Medicare kind of events, like how you code this cancer diagnosis based on different codes, and different algorithm will give you different results. So for this study, what they did was actually look at an overlap period. They're trying to compare what's happening in the trial and what the diagnosis is in Medicare, and to see how good they are, how good those Medicare diagnoses are. I would say it's okay performance, but it's not like, of course, not 100%. There were maybe 80% like a concordance. So yeah, but that's also like an issue. Like we should address, but we haven't.
Okay, so if there's no more question, let's thank Dr. Zhao again for the.